Thank you, Ralph, and John, for the invitation to come talk to you today. And thanks to all of you for coming here so early in the morning. So I'm going to tell you about my lab's work to develop tools for imaging the electrical activity in neurons in real time. To study a neural circuit, one would like to record the activity of a large number of neurons with high resolution in space and time without perturbing this activity. We've developed a protein which you can express in a neuron, and when the neuron fires action potentials, these are accompanied by flashes of fluorescence that you can readily see in a microscope. This is real data. This is a single trial recording from a neuron taken at 1,000 frames a second. And that's basically all I have to say. So that's the whole summary. Okay? But before I take questions, I'll go into a little bit more detail about this. And by way of disclosure, I've started a company which is working on making this technology more broadly available. All right. So this is an artist's conception of what we'd like to do. We'd like to make neurons light up when they fire so that we can see global patterns of activity. The signals we're after are action potentials, which are about 100 millivolts high and last about a millisecond. The challenge is that if you just look at a neuron, there's no endogenous contrast to give you an optical signal that you can see. Just like if I look at this wire, I can't see what the voltage is in the wire just with my eyes. And so we need some sort of a contrast agent. And for decades, people have been developing contrast agents, both small molecule and protein-based, to try to convert these changes in membrane voltage into an optical signal. And the challenge has been to find something that's sufficiently fast and bright and sensitive enough to do this without perturbing the activity of the neuron, and that has enough resolution so that you could see this in tissue. Great. Most of the work that I'll tell you about is going to come out in about 2 weeks. So the story that I'll tell you starts here. This is a view from an airplane looking down. Does anybody know where this is? That's right. This is the base of San Francisco Bay. This is coming into San Francisco Airport. And if you look down from an airplane, you can see these are salt ponds where people are evaporating seawater to make salt. And you see these beautiful colors in the water. To see where these colors come from, we can zoom in by 12 orders of magnitude. And what we see is that there are microorganisms growing in the water, halophilic microorganisms, that are producing these proteins called microbial rhodopsins. These are transmembrane proteins with a covalently bound chromophore of retinal in the core of the protein. And it's this chromophore of retinal which gives the color that you can see from the airplane. There are thousands of varieties of these proteins known in the wild, and they mediate a huge variety of interactions between sunlight and biochemistry. So for instance, there are light-driven proton pumps, there are light-driven chloride pumps, there are light-activated ion channels, and light-activated signaling molecules. Now, a neat feature of these proteins is that they come in all the colors of the rainbow. And this is a little bit surprising because they all have exactly the same chromophore in them. It turns out that the opsin, the protein around the chromophore, can tune the electronic energy levels to shift the absorption spectrum throughout the visible spectrum. In fact, this is the same color tuning mechanism which we use to have color vision in our eyes. And so in a sense, the color tunable range of these proteins defines the visible spectrum, because that's the same set of physics that we use for our vision. Now, almost 10 years ago, people realized that you could take the genes for these proteins and put them into other cell types and thereby gain optical control over various processes in these other cells. And most famously, if you take channel rhodopsin and put it into a neuron, then by shining blue light on the neuron, you can depolarize the cell and trigger it to fire. And this has been the basis of a huge amount of important work in neuroscience, and it's called optogenetics. So here we have these proteins where you can shine light on them and use the optical signal to induce a change in membrane voltage in a cell. So light comes in, and a change in membrane voltage comes out. A few years ago, I said to myself, I wonder if we can run these things in reverse. Instead of having light come in and a change in voltage come out, can we use a change in the membrane voltage in a cell to induce some conformational change, which will maybe lead to a color change or some spectral change, which we could see in the microscope. Okay? So we want voltage in and an optical signal out. Great. So now I'm going to skip about 3 years of really hard work and tell you that we found a way to do this. The protein that we work with is called Rhodopsin 3, and it's derived from a microorganism that lives in the Dead Sea. In the wild, this protein acts as a light-driven proton pump. So it absorbs sunlight and pumps a proton from the inside of the cell to the outside of the cell. Its host then uses that voltage to provide part of the proton motive force to power its ATP synthase, so to make a living from sunlight. We discovered that this protein is a little bit fluorescent. It's a new class of fluorescent proteins with no homology to GFP or any other known fluorescent protein. But it's only fluorescent when there's a proton sitting on a particular functional group right here in the middle of the protein. A change in the voltage can pull that proton on or off of that functional group. So the voltage can tune an acid-base equilibrium inside of this protein. And only the protonated state is fluorescent. So the voltage tunes the fluorescence of the protein. This is a picture of a hex cell expressing wild-type Rhodopsin 3, so the protein straight out of this creature in the Dead Sea. And you can't see it here, but there's a little patch pipette jammed into the back of the cell. We're looking at the endogenous fluorescence of this protein. And I'll show you a movie of what we see when we apply steps of voltage between plus and minus 1/10 of a volt. This is real-time raw data. So what you should see is when the voltage goes up, the protein gets bright, and when the voltage goes down, the protein gets dim. Okay? Great. So now I'll tell you a little bit about the spectroscopy of this protein. We excite it with orange or red light, and it emits in the near infrared. This is very convenient for biological imaging because these are wavelengths of good tissue penetration and very low phototoxicity and autofluorescence. The quantum yield is terrible. For our best mutants, it's about 1 or 2%, so 50 or so fold dimmer than GFP. But it turns out that these proteins are extremely photostable. Here I'm showing a photobleaching trace of a microbial rhodopsin and a typical organic dye under the same illumination intensity. And the dye photobleaches in about 30 seconds, while the rhodopsin lasts for about 30 minutes for the same amount of photobleaching. So even though these things are dim, because we're exciting them so far into the red, we can turn up the laser power and still get reasonable signals out of these things, even without too much photo damage or photobleaching to the sample. Now, one of the problems with a wild-type protein is that it's a light-driven proton pump. So when you shine light on it, it generates a current which perturbs the membrane voltage. Here you see, as we turn the laser on and off, we get this photocurrent in a cell. And that's undesirable because we're trying to make a sensor and not an actuator. It turns out there are many ways to break the protein. And so you can mutate the proton pumping pathway to get rid of this photocurrent so that it doesn't perturb the membrane voltage anymore, but still preserve the voltage sensitivity. So working with Robert Campbell's lab at the University of Alberta, we did a directed evolution screen and made some new versions of these proteins, which we call Quasars, for quality superior to Arch, which show better voltage sensitivity and are brighter and don't generate a photocurrent. So one of the figures of merit for a fluorescent voltage indicator is its sensitivity. How much change in fluorescence do you get per change in membrane voltage? To measure this, we just apply triangle waves of voltage to the cell. So this is what the raw data looks like. And you can see for this mutant, we get about a 2-fold change in fluorescence over the voltage swing of a typical action potential. Another important figure of merit is the speed of the response. So if we have a step in voltage, how quickly does the fluorescence respond? And here's what the raw data for that experiment looks like. For the wild-type protein, the step response is about 600 microseconds. For this quasar variant, most of the response is about 1.2 milliseconds, and then there's about a minor component which is slower with about 15 millisecond time constant. We have another variant of the protein, quasar 1, which has a step response of less than 50 microseconds. So these proteins respond extremely fast to changes in membrane voltage. So between the high sensitivity and speed of these proteins, we thought that it was plausible that they could report action potentials in mammalian neurons. To test that, we expressed these proteins in cultured rat hippocampal neurons and used a patch pipette to inject current pulses to induce action potentials. And then we recorded the fluorescence at the same time as the voltage. And so here's what the raw data looks like. The blue trace is an electrical recording of these action potentials in a neuron. And the red trace is a single trial simultaneous fluorescence recording taken at 1,000 frames a second. And what you should see is that there's a correspondence between these two traces. Here's a close-up of the data, and you can see the close correspondence of the optical recording and the electrical recording. This is with no filtering or processing other than just averaging over the whole cell. Okay? Because we're doing this in a microscope, we can take videos of what these action potentials look like. So here is a neuron, and you can't see it, but there's a little patch pipette jammed into the cell. And I'll show you a movie at 1,000 frames a second of what an action potential looks like in a neuron. This is averaged over about 100 repetitions of the stimulus to improve the signal to noise. Right. So that's what a neuron looks like when it fires. Now, this is a single cell, and we wanted to look at big networks, to look at larger numbers of neurons. So towards that end, we've developed a new microscope apparatus, which we call in the lab our Can Microscope, because the objective is about the size and shape of a soda can. So it uses a giant objective to image a very large field of view with very high light collection efficiency. And then we have some custom optics and electronics for recording these large fields very quickly. With this, we can now image large collections of neurons. So here's a field of view, which is about 1 millimeter by 4 millimeters. There's maybe 50 or so neurons in this field of view. These are, again, cultured rat hippocampal neurons. They're synaptically connected to each other. And in this circuit, we see spontaneous activity. They're expressing this quasar voltage indicator. And here I'll show you a movie at 500 frames a second, slowed down tenfold of what this network activity looks like in this neuronal culture. So here you can see every neuron, you can see every action potential as they flash, and you can also see the subthreshold voltages in the neurons as they light up. Okay? Great. Now we've been interested not just in looking at voltage in neurons, but also in other systems. You know, everything with a membrane could, in principle, support a membrane voltage, and we're interested in exploring the diversity of bioelectric phenomena. Now, one place where membrane voltage is obviously important is in the heart. And so we've been expressing these reporters in human-induced pluripotent stem cell-derived cardiomyocytes. So you can take some skin from anybody and reprogram these skin cells back through embryonic development into induced pluripotent stem cells, and then take them forward through development down a different pathway, for instance, into cardiomyocytes. And if you culture these cardiomyocytes in a dish, they'll start to beat spontaneously. And you can do this either with cells derived from a healthy person or derived from somebody with a genetically based cardiac disease. And one of the challenges in using this technology for drug discovery and disease modeling has been the difficulty of recording the electrical behavior in these cells. With these optical measurements, we can now look at hundreds or thousands of cells in parallel, and so we can do fairly high throughput measurements of electrical activity in cardiomyocytes. So here's what a human cardiomyocyte looks like when it expresses one of these indicators. It's beating spontaneously in the dish, and so you can see it light up as it beats. And here's a plot of the fluorescence of one of these cells recorded for 1,000 seconds continuously. And you can see that it's beating at the beginning and it's beating at the end with no sign of phototoxicity or photo damage. And there's about 30% photobleaching over this 1,000-second recording from a single cell. Now, in the heart and also in neurons, the electrical impulse is then triggers a calcium response. And in the heart, the calcium response is what actually makes the muscle contract. And so we thought it would be nice to look at the voltage and calcium at the same time. To do that, we just stapled a genetically encoded calcium reporter to our genetically encoded voltage indicator. And so we call this construct Caviar for calcium and voltage indicator. And the key point here is that the calcium indicator is based on GFP, and so it's excited by blue light and emits green, while our voltage indicator is excited by red light and emits in the near infrared. So you can look at both of these at the same time. Here's a human cardiomyocyte expressing this Caviar construct. We're looking at the voltage on top and the calcium on the bottom. And this construct is, the cell is beating spontaneously. And so you can see both signals. And if you look closely at the calcium signal, you can see these little flashes here. Those are called calcium sparks, which correspond to individual puffs of calcium being released from the sarcoplasmic reticulum inside of the cell. And so here's a plot of the voltage and the calcium signals from the cell. And you can see the electrical signal is actually more stable than the calcium signal in terms of photobleaching. So we've been using this now for drug screening, and I'm not going to go into this in much detail. I'll just show you an example of what the data looks like. So these are recordings from single human cardiomyocytes where we've added different compounds. So these are recordings taken at different concentrations of the compound. B.R.A.I.N is a drug which is used to make the heart beat more strongly. So as you add B.R.A.I.N, you can see the amplitude of the calcium signal increases. And in the heart, that would make the muscle contract more strongly. Nifedipine is an L-type calcium channel blocker. So as you add the nifedipine, you can see it blocks the calcium current. And in the heart, the calcium current prolongs the action potential. So when you block the calcium current, you can see the action potential speeds up. And dofetilide is a compound which blocks the delayed rectifier potassium channel, which is responsible for the repolarization of the heart. And you can see as you block that channel, you get delayed repolarization, and you even get these early after depolarizations, which prolongs the action potential interval in these cardiomyocytes and also affects the calcium signaling. So by looking at these traces of voltage and calcium, you can read off the mechanism of action of these drugs and see how they affect cardiac activity. We're also interested in looking in live animals. We've made a zebrafish, which expresses this calcium and voltage indicator in its heart. So now we have caviar in fish. Here's an image of the calcium signal in the heart of this fish. So you can just take the whole fish and plunk it down on the microscope, and you can see the calcium signal in its heart. We've developed a custom spinning disk confocal microscope for doing optical sections inside of the live animal. So here's a video of the electrical signal in the heart of this fish. This is taken inside of a live fish. Fish have a two-chambered heart, so here's the atrium and here's the ventricle, and you can see the electrical signal propagating through the fish. Whoops. We've written software to stitch many of these sections together to create a three-dimensional map of the electrical activity in the heart inside the live fish. And now we've done this as a function of time through embryonic development to see how this organ boots up. How does the electrical activity arise and then develop through development? And then we've been using drugs to block different channels at different points to see which channels are responsible for mediating the action potential at different times and in different chambers as a function of embryonic development. We've also been using this same technology now to look at voltage and calcium at the same time in the heart of the fish. And so here's an optical section, and then you can see as we change the focus, we'll move up and down through the heart of this live fish. We're working on doing this in the brain of the fish, but we haven't gotten that to work yet. Great. This calcium and voltage indicator also works in neurons. So this is a cultured rat hippocampal neuron. And here you can see the optical recording of the voltage. And here's the simultaneously acquired optical recording of the calcium. And this really emphasizes the difference between voltage imaging and calcium imaging. So in the voltage imaging, you can see every action potential. In the calcium imaging, you can see isolated action potentials, but when you have a big cluster of them, it's hard to detect the individual events. Subthreshold events, which don't manifest as action potentials, have no correlate in the calcium signal. All right. Now, so far, I've been showing you passive recordings of the electrical activity in cells, or we've been using a patch pipette to induce electrical activity. But using a patch pipette to trigger a cell partially defeats the purpose of having this optical recording, because we'd really like to do things without electrodes at all. Now, at the beginning, I mentioned this gene channel rhodopsin, which is a blue light activated ion channel. You can shine blue light on it and trigger a neuron to fire. So I thought, wouldn't it be nice to combine the channel rhodopsin blue light actuator with the archirhodopsin-based red light reporter in the same cell? That way we can trigger the cell to fire with blue light, and we can record from it with red light. So we can do all optical electrophysiology. This is a collaboration with Ed Boyden's lab at MIT. So we developed a construct to do this, and the idea is we're going to flash blue light on a cell and then see it respond with a flash of red fluorescence, just like that. And you see this works in PowerPoint, and so that's very encouraging. Here's some real data from this construct. So each blue bar here represents a flash of blue light delivered to a neuron. The red is a fluorescence recording, and the black is a simultaneously acquired electrical recording. This is single trial data with no signal averaging. Here's a close-up of these traces, and you can see again the correspondence of the optical trace and the electrical trace. So here we're using the patch clamp for ground truth, but it's not necessary for the experiment. Now, one of the neat things about having an optical stimulus is that you should be able to shine the light in any pattern of space and time that you like in order to trigger maybe a subcellular region of a neuron or one neuron within a complex circuit. We made a machine to do that based on a digital micromirror device. So this is a hacked version of a video projector, just like the one that's displaying the image on the screen here. Instead of projecting an image onto a video screen, we use this digital micromirror array. It's an array of about 2 million individually addressable microfabricated mirrors to modulate the blue laser beam on its way into the microscope. And we re-image the micromirror array onto the sample in the microscope. So we can project an arbitrary pattern of light, arbitrary in space and time, onto the cells in the microscope. Then we use flood illumination with red light to record the activity in the whole field of view. Just to demonstrate that this works, I took a fluorescent film, just fluorescent plastic beads, and projected a video from YouTube onto these beads inside the microscope, and then recorded the view inside the microscope. And so that's what this looks like. So this is a video of a clown taken inside the microscope. And a single cell would be about this big on this field of view. Okay? So you can really stimulate in any pattern of space and time that you like. We've used this to look at synaptic transmission in cultured neurons. Here's an example. Here are 2 neurons in culture, which are synaptically coupled. We've artificially colored one green and one blue. They're both expressing this optopatch construct. We're going to optically stimulate just the cell body of the one on the left. And what you'll see is that this will induce a series of action potentials in the cell on the left and an excitatory postsynaptic potential in the cell on the right. This movie is, again, averaged over about 100 repetitions of the stimulus. And this is a video at 1,000 frames a second. It goes by quickly, so you have to pay attention. So there's some action potentials here and an excitatory postsynaptic response over there. To check that this really is synaptic transmission, we then added synaptic blockers. And now you'll see that the upstream cell will continue to fire action potentials in response to stimulus. But if you look at the cell body of the downstream cell, there will be no response. So this provides a comparatively high throughput way to study synaptic transmission in cultured neurons. Great. We've also implemented this in the wide field system. Is there a way to lower the lights on the screen? Maybe yes, maybe no. OK, anyways, if you can't see it, just imagine lots of lights flickering. So here's a wide field image of, again, this is about 1.5 millimeters by 4 millimeters. There's about 50 neurons here. We'll optically stimulate the whole field of view, and you'll see the neurons start to respond. This is a video at 500 frames a second, slowed down tenfold so you can see it. When the blue light goes on, there'll be a little icon here which shows up. Right? So this is a tenfold slowdown of what's going on inside here. This is single trial recording, and you can see the neurons responding to this optical stimulus. And you can record every action potential in every cell in the field of view. If you zoom in and look at any one of the cells, this is what the response looks like. Here, we're stimulating with successively higher powers of blue light, and you can see as we stimulate more and more, the cells fire more and more. But each cell has a unique firing pattern in response to these increasing stimuli. And so this provides a means to characterize with fairly high throughput the excitability of all the cells in the culture. You can also use the micromirror array to go in and stimulate any one of these cells or any combination of the cells in any pattern of space and time that you like, and then look at the response of every cell in the circuit. So for cultured neurons, this comprises a complete neuroelectronic interface where you can stimulate in any pattern that you can conceive of and record every event in every cell in real time. Great. Now, one of the challenges that we face with this technology is that these action potentials are just so fast. They only last about a millisecond. And we have the best cameras that money can buy, and they can go at 1,000 frames a second. So they have the time resolution to detect these action potentials, but not really to map the details of the subcellular propagation through the dish. And we wanted to image with much higher time resolution. One of my students came up with a very clever trick for imaging, for mapping the propagation of the action potential wavefront with higher time resolution than the exposure of the camera. And here's how it works. We take a series of images of a neuron firing at 1,000 frames a second. And then for each pixel, you can plot the fluorescence as a function of time during the action potential. And so the fluorescence goes up and comes back down. And we're sampling this at discrete intervals corresponding to the individual exposures of the camera. And so we get this discretized map. But we know a lot about what the underlying waveform of the action potential looks like. In particular, we know that it's a smoothly varying continuous function. And so we can interpolate this waveform through these discretely sampled points, and then infer the timing with which this interpolated waveform crosses a user-selected threshold, say 50% of the peak, with much better time resolution than the exposure time of the camera. So you can think of this as a model-based fit. This is qualitatively similar to a time domain version of the point spread function fitting, which is the basis for super resolution imaging in the spatial domain. Okay? So we do this fit independently at each pixel in the movie. And then we make a new movie where we light up each pixel only at the time corresponding to this interpolated timing. So we can make a movie showing the propagation of the wavefront through the sample. And this actually works. Here I'll show you a movie where we're optically stimulating a neuron here using the micromirror device. There's no electrodes here. And first I'll show you the movie at the native frame rate of the camera, 1,000 frames a second. So we stimulate it, and we see this flash corresponding to the action potential in the cell. But the flash only lasts about 2 or 3 frames, and you can't really see the propagation through the cell. Now I'll show you the same data where we've interpolated the timing to 20,000 frames a second, so 50 microseconds a frame. And here you have to remember we're processing each pixel independently, so we don't impose any wave-like motion on the data. And so here's what we see. So you can actually see now how the signal is propagating through the neuron. Here I'll show you another example where we're stimulating the cell body here. And this is interpolated to 50,000 frames a second. Okay. And so you can really see in detail how these signals propagate through a single cell in culture. Now, if you look closely at this, and I'm not sure if the contrast really shows this, it looks like the action potential is starting not where we stimulate, but a little bit over here. And again, the screen doesn't show it, but there is a very skinny piece of spaghetti over there coming out of the cell body. Here's another example. Here we're stimulating not the soma, but we've just drawn a patch over here, and we're going to stimulate some dendrites. This is, again, I think at 50,000 frames a second. And you can see the action potential propagate through the cell. And again, if you look closely, you can see that the action potential is starting over here, not where we stimulate. And so what's going on? Well, in a neuron, the neuron takes dendritic inputs and integrates them. And our belief is that these inputs get integrated at the axon initial segment, where the axon joins the soma, where the threshold for action potential initiation is lower than elsewhere in the cell. And so we would expect that the action potential should start at the axon initial segment and then propagate down the axon and back into the dendrites to both the ones that are stimulated and the ones that are not stimulated. And so looking at these cells in culture, it's hard to know what the axon is. But we hypothesized that this little region right here, where the action potential was starting, was the axon initial segment. To test that, we then immunostained this cell for ankyrin G, which is a marker of the axon initial segment, and then came back and found the same cell in the microscope. And so here's an overlay of the immunostaining data. The red is the anchor G marker for the axon initial segment. And you can see indeed that this biochemical marker corresponds to the functional region of action potential initiation. And so we can correlate the details of action potential propagation that we see with the optopatch technique with the underlying biochemistry of the cell. Great. We've also been using this in whole tissue. And this is more preliminary work, but we've expressed this optopatch construct in organotypic brain slice, also now in acute brain slice with AAV gene delivery. And we can optically stimulate single cells in the slice and record firing patterns of individual cells in the slice. We haven't yet been able to do very wide field recordings because it takes too much laser power. All right. So I've been showing you mostly mouse and rat neurons. We're also interested in looking at human neurons. And so working with Kevin Egan, we've been trying to develop a pipeline for taking human cells, so fibroblasts from real people, and converting them into induced pluripotent stem cells, and then taking them back through embryonic development to convert them into neurons, which can start to fire in the dish, and then to do optical electrophysiology on these cells to characterize them and to try to learn something about what's going on in the patient. And the idea is that this can give us a way now to study the function of human neurons from real people, either from healthy people or from people with genetically based diseases of the nervous system. And so we've done a proof of concept experiment to test this pipeline with a cell-based model of ALS, a neurodegenerative disease, a disease of motor neurons, to see if we can do this. And so ALS is it's degeneration of motor neurons. And so in Kevin's lab, they've made human stem cell-derived motor neurons. These neurons had a mutant in the SOD1 gene, a single point mutant, which is a highly penetrant risk factor for ALS. In Kevin's lab, they also edited the genome of the stem cells to correct this mutation to make another population of cells that are genetically identical in every way except for this single mutation. So this way we can study the effect of the mutation in the same genetic background. They made these into motor neurons and then gave them to us blinded. We didn't know which ones were which. And we delivered this optopatch construct and measured the excitability of the cells. So we stimulated them with patterns of blue light and looked at their firing as a result. Now, Kevin had previously observed that in the neurons with this mutation, there's a hyperexcitability phenotype. The cells fire more in the absence of stimulation. This correlates with a clinical observation that patients with ALS have hyperexcitable motor neurons. And clinically, the greater the excitability of the motor neurons, or in the patients, the worse the prognosis for the disease. Kevin recently found a drug which reverses the excitability phenotype in the dish, and now they're going into a clinical trial to try that drug in patients. So that's the backdrop to this experiment. So we wanted to see if we could reproduce that optically. The work to figure out this phenotype originally had been done by manual patch clamp, and it took about 2 years of manual patch clamp to get this data. So we culture these cells and stimulate them with light and record their flashes. So here's what the raw data looks like. Now, one of the challenges here is that these cells tend to snuggle up against each other. And so there's quite a few cells here, and we have to disentangle the signals from each of the individual cells. We use a technique called independent components analysis to identify the clusters of pixels that are firing in synchrony and firing independently of the other clusters of pixels so that we can disentangle these overlapping signals. This technique was developed by Eran Mukamel, who's sitting somewhere in the audience here. And so out of this movie of these overlapping cells, the computer finds these clusters, which look like the individual cells, and each cluster has a unique firing pattern. Now, this data points out something important about this kind of measurement. These cells are all genetically identical, and they're all cultured in the same cluster. So they're as close to identical as you could possibly hope for. Yet you see very different firing patterns. And this really emphasizes why it's important to have high throughput measurements for optical characterization, or for characterization of the cells. There's a lot of variability in these cells, and you really need statistics to overpower that. So we took recordings like this for a couple of hundred cells with the ALS mutation and a few hundred without the mutation. Then for each cell, we found all the spikes, so every time the cell fired. And then we made a matrix where each row here corresponds to data from a single cell. I don't know if you can see it. There's white spots on this matrix, and each white spot corresponds to one firing event of the cell. Okay? And so as we stimulate them more and more, you get more and more white spots. And we lined up all the cells with the mutation and all the cells without the mutation. And then the question is there a difference in this pattern of white spots from this pattern of white spots? Is there a difference in the firing? There's many ways to analyze this. The simplest one is just to average across all the rows to ask what's the mean firing rate as a function of time for the stimulus in the cells with the mutation and without the mutation. So we did that. Here, the red is the mutant. The black is the genome-corrected control. And here, as a function of time, you can see that the cells fire more as we stimulate them harder. But you see that in the mutant cells, there's a higher firing rate at baseline. So when we're not stimulating the cells, the mutant cells are hyperexcitable. They fire more. But also, when we stimulate hard, the mutant cells actually show less activity. They inactivate. And this is consistent with sodium channel inactivation in these cells. And so this gives us a phenotype for the disease, which we can then use both for mechanistic studies and potentially for drug screening. Acquiring this data on a couple of hundred cells only took about a day, in contrast to the 2 years of data acquisition that it took to get the initial phenotype. All right, so I'm going to finish just with a comment about where this all comes from. It's really incredible to me that we can use genes taken from microorganisms that live all over the world and just staple these things together to come up with new functions which have never been selected for in evolution. So our Rhodopsin voltage indicator comes from this creature which lives in the Dead Sea. Our Rhodopsin, it's a new one which we developed with Ed Boyden. It comes from a microorganism, an alga, which lives in a freshwater pond in the south of England. And the Rhodopsin is fused to a fluorescent protein derived from a coral, and the Rhodopsin is fused to GFP from a jellyfish. And then these things are actually linked by a sequence from a pig virus. And so you can just take all of these things and staple them together. And the only reason we can do all of this is because ecologists have been going out for decades and characterizing all of these seemingly esoteric creatures living in the ocean. I think the Israeli ecologists who found Rhodopsin in the early 1980s never would have guessed that this protein would one day make its way into a cell-based model of a human neurodegenerative disease. All right, so to summarize, this is a picture of the Dead Sea where our protein comes from. I've told you about these Rhodopsin-based voltage indicators. I told you about a combined reporter for calcium and voltage. And I should say, calcium here really is a proxy for any GFP-based reporter. So any physiological quantity that you want to measure in parallel with voltage, you can. So there's reporters for cyclic AMP and ATP and pH and so on, and you can stick any of those there. I told you about this optopatch trick for optical stimulation and recording from cells, and about this sub-Nyquist action potential timing algorithm for inferring propagation with very high time resolution. And now that we have these tools, we're applying them every place we can find a membrane. So today I focused on neurons and told you a little bit about cardiomyocytes, but we're also using these to study stem cells and stem cell differentiation. We're looking at bacteria, and we're looking at embryonic development. I've worked with many great collaborators on this project. I particularly want to mention Joel Kroll, who was a postdoc who started this. He really bet his career on this and started from when this was just a crackpot scheme. He's now working at our company. Adam Douglas, who's now at the University of Utah, and Daniel Hochbaum, who's a grad student in my lab, took all the initial work data in neurons. And Daniel really developed this optopatch technique and did most of the experiments that I showed you today. I've also had many great collaborators at many different institutions. These people pay the bills. This is a photo of my lab, and I'm happy to take questions. Thank you. Can we have the lights? Yeah.
So Lauren Luger started out with GCamp1, as you know, I don't know how long, but 10 years ago, and using a pipeline at Janelia, which I understand was a $10 million investment, was able to get that up to GCamp. Now it's like 7 or 8,
6,
which has vastly improved the sensitivity, the time course, and so forth. And I wonder if we gave you $10 million, how much better could you do?
Well, we can try the experiment. It's a good question. So I should say there are many people working on voltage indicators. There are people working on small molecules. So Roger Chen's lab recently came out with a very promising organic dye. And there are several labs working on protein-based indicators. And so it's a horse race right now, and there's different strategies. I was actually just at Janelia Farms reviewing their GCamp project, and they're now starting to think about looking at voltage indicators as their next target. I think an interesting question, to which I don't know the answer, is whether the right approach is to throw a ton of money at one group and to really have them just crank on it, or to spread that around and to have lots of people try different things. I would say I'm not certain that any of us has found the right scaffold upon which to optimize to get the best indicator. And I think for different applications, there will actually be different indicators because they all have their sort of peculiarities. At this point, for the Rhodopsins, it is a question of optimization. And in my lab, we're thinking about ways to do now high throughput screening of the functional attributes of these cells in order to improve their performance. I hope we can do it for less than $10 million. Yeah. Yeah.
So I was excited by your approach to dilating time. And I wondered if you'd tried that yet with the Caviar probe in the heart and the zebrafish, because there should be some fine structure to that
Yes.
propagation, both the calcium and the voltage.
Mm-hmm. Yeah.
And then second piece of that question, because I won't get the microphone again. Have you tried putting this probe in close association with one of the key channels, like the sodium channel?
Okay. Both great questions. So we have applied this sub-Nyquist action potential timing in cardiomyocytes, both in culture and the zebrafish. In the zebrafish heart, if you do this on the voltage channel, you can then watch the propagation from the atrium to the ventricle, and you can see in great detail how the signal propagates through the heart of the fish. In cultured cardiomyocytes, you can, so we haven't done it to the calcium signal in the zebrafish heart. We have done it to both the voltage and the calcium in cultured human cardiomyocytes. And with the calcium signal, it's pretty neat. You can see the calcium waves propagate out from different foci in the cell as you get calcium-induced calcium release spreading through the cardiomyocyte. And so that's something we've done a little bit of, but we haven't explored in detail. The second question was whether we've tried associating these reporters with particular channels, any of the channels. And we have not done that. It's not clear to me how we would get good intimate association of the reporters with the channels. One of the big challenges for us, which continues to be a challenge, has been the trafficking of these proteins. Even getting them to the plasma membrane is not something to take for granted. And part of the 3 years of work that it took to get our first signals involved trying to find the right backbone which trafficked efficiently to the plasma membrane at all. So for instance, we've tried targeting these things to mitochondria to look at mitochondrial membrane voltage, and that has not worked. We haven't been able to get them to the mitochondria. Yeah.
Just a couple questions. One of them is, what are the energy requirements for evoking the images, and what are the prospects for doing this in vivo in the future? What are the challenges there? And the second minor question is just, I was looking at the traces for intracellular voltage and the quasar image in the experiment with Ed, and there seemed to be some nonlinear relationship, and I wanted to know if you'd explored that and understood what it was about.
Great. So the first question is about going to intact tissue and in vivo. So we typically image at an illumination intensity of 300 to 800 watts per square centimeter. And so that's pretty high. But you have to remember it's red light, and so most of the light just goes right through. This is in culture. Most of the light just goes right through, and so we don't see any effect of that. So we've been doing recordings in zebrafish heart, and with a focused laser beam, that turns out to be not too much power. That's a few tens of milliwatts if you want to look at single cells in tissue. And I think looking at single cells by vivo, I assume you mean in a mouse, is within the foreseeable future. To do wide field recordings in mouse, or well, really in any intact brain, will, I think, require a qualitative improvement in the brightness of these things, because right now it's just too much laser power. We've tried looking in flies and fish, and we're continuing to work on that, but I am very much concerned about the amount of laser light that we're putting in. And the second question was?
Oh, just the relationship between voltage.
Oh, right. The second question was about the relationship between voltage and fluorescence. So the relationship is pretty linear, as I'll show. So here's the plot of, oops, this is the wrong slide. There we go. Here's the plot of fluorescence as a function of voltage, and you can see that it's pretty linear. In cases where we see a discrepancy between the voltage and the fluorescence, it usually looks like the error is actually in the patch clamp recording, and it comes from improper compensation of the series resistance in the patch pipette. One of the concerns with these things, of course, is about whether we're perturbing the electrical behavior of the cell by expressing these things. And we've checked membrane resistance, membrane capacitance, resting voltage, threshold voltage, and action potential waveform, and we don't see significant effects there. Of course, if we overexpress reporters, you can see an effect. I think another concern, which probably doesn't get enough attention, is what is the perturbation from patch clamp. So when you jam an electrode, or when you jam a patch pipette into the cell, you dialyze out a lot of the components. And we see, so if you look at a cell optically, and then you patch it, and then you look at it optically again, you see perturbations from the patch pipette. Yeah, Roger.
Okay. How much gating charge is introduced by these sensors, and do you have to worry a little bit about how much expression level, and also at the beginning there was a lot of concern that these were multiphoton processes intrinsically, and I haven't heard anything about it today, but it originally was stated that there was a multiphoton process which did the sensing, and has that changed?
Great. So the first question is about expression artifacts and capacitive loading of the cells.
Capacitive loading, yeah.
So we've measured the capacitance of cells expressing and not expressing, and the capacitive load is very, very small here.
Oh.
And let me just expand on that a little bit.
Yes.
So with many voltage sensors, there's a problem that in order for this thing to sense voltage, it has to shift charge across the membrane, and something has to move in response to the voltage, and that then leads to a capacitive artifact, which can suppress firing and can affect the electrophysiology of the cell. One way to get an insight into how much charge is moving is to look at the shape of the fluorescence versus voltage curve. If you have something which has a very steep sigmoidal dependence, that means that there are many charges moving in synchrony across the membrane. And basically what happens is because there's many charges moving together, the energy change is large enough that it overwhelms KT with just a small shift in voltage, and so you get a big capacitive artifact. We've calculated the gating charge in these based on fitting a sigmoid to these curves. And it turns out to be about 0.1 electrons as the gating charge, because the charge only moves a small fraction of the way across the membrane. And the capacitive load is proportional to that gating charge squared. And so that turns out to be a very small effect. On the other hand, we are expressing at a high level, and the capacitance is proportional to the expression level. And so that is a concern, but we've tested experimentally, and it seems to be quite small. If you overexpress these proteins, you certainly can kill the cells. And so we see that there is a sort of therapeutic window where we can get enough expression to see activity, but not so much that we kill the cells. We're using the same promoters that have been used in transgenic animals to express these same proteins, and these animals seem to run around and be okay. And so at least, I'm sure there are effects at a subtle level, but they seem not to be very significant. And the second question was? Multiphoto, thank you. Yeah, that's an important question also. So the wild type arch, and I didn't get into this, the wild type arch has a very complex photocycle. And we found that the fluorescence and the voltage sensitivity depended on absorption of 3 sequential photons. And so this was a big issue because it meant we really had to shine a lot of laser power on the cell in order to get a signal. We've characterized the nonlinearity in these mutants, and it's gone. And so they behave more like conventional fluorophores. And so part of the directed evolution screen, that wasn't exactly what we were selecting for, but by selecting for brightness, that as a side effect, that got the fluorescent state to be the ground state of these proteins. Yeah, so the nonlinearity is no longer an issue.
Why did you pick the Dead Sea? It's really unique as a source of water.
Why did I pick the Dead Sea? Oh, well, I didn't pick the Dead Sea. The story is we were working with a different protein derived from a different microorganism, which happened to be from Monterey Bay. And we spent about a year trying to get that protein to work in mammalian cells, and we couldn't get it to traffic to the plasma membrane. It just didn't work. Then Ed Boyden's lab published a survey of many different microbial rhodopsin proteins, trying to use them in the forward direction as optogenetic tools. And they surveyed genes from all over the world. And it turned out that this archaeorhodopsin one was the most effective one in their survey. And so we knew from that that this protein trafficked to the plasma membrane because they tested it in mammalian cells. We didn't know whether or not it was a voltage indicator, but we knew that it had solved the one problem which was holding us back. And so then, based on that, we asked Ed for the gene for this protein and tried running it in reverse, and that just worked on the first try. So before getting to that, we had tried 45 different constructs with a prior protein, all of which had failed.
Quick question. Can you comment on the instrumentation part? So if your voltage sense dyes are all perfect, and I see you have done a lot of wide field imaging and all those stuff. So for genome project, there was a sequencer that made this thing all happen.
Yeah.
So for your optical imaging, what is the really essential thing for dream wish list you have?
Yeah. So the question is about the instrumentation. We use different instruments for different experiments, and so there is no one-size-fits-all solution. The most basic version is a inverted epifluorescence microscope with a high NA, either oil or water immersion objective, a laser, and either an EMCCD or a scientific CMOS camera. So it's similar to the kind of setup that you might use for a single molecule fluorescence imaging experiment. That's sort of the level of sensitivity that you need for this. And unfortunately, there is no commercially available instrument that has exactly those attributes, but it's not hard to put one together. For the wide field experiments or the experiments in tissue where we're doing optical sectioning, that's much more specialized instrumentation. And so there we have to use some custom optical elements, and I could go into more detail about exactly how those things are put together. That was part of the motivation for starting a company also, is that to make the technology more broadly available.
So what is it going to take to see minis? Is it just a matter of getting the dye to the right place, or is there some other limitation to see these much smaller?
Oh, to see MINs, you said?
Yeah,
Minis.
mini implant potential, yeah.
Yeah, well, so we can see subthreshold events. The signal-to-noise ratio in the, when everything is working right, is about 2 or 3 millivolts of precision in a 1 kilohertz bandwidth. So that means if you integrate for a millisecond, you can see a voltage change of 2 or 3 millivolts. And so you can definitely see subthreshold events. We've compared simultaneous patch clamp recordings and fluorescence recordings in synaptically active cultures, and we can't quite see the minis yet. I think a factor of 2 or 3 in either expression level or ideally in brightness would be sufficient to see minis.
Hi. With the use of high-power lasers, do you see any temperature changes?
Great question. So you might have noticed that the laser power we were using for those wide field images was 3 watts of laser power, which is a lot of laser, right? But don't freak out. It's spread over a very big area. And the reason we do this is to get high intensity over a large area takes a lot of laser power. And most of that light does go through. We have quantified the temperature change because, of course, this was a huge concern. And it does raise the temperature by about 4 degrees in these samples. And so we typically run at, so if we don't heat it all, if we're nominally at room temperature, that will raise the temperature to about 30 degrees. And that's just through absorption in the water. So it doesn't matter whether or not the cells are there. Yeah. Great. All right. Thank you.